Everyone, so in today's class we are going to learn about the protein structure. As most of the classes start from the biomolecules and they start teaching about the primary, secondary and tertiary as well as the quaternary structure, but first you need to understand the basics. So once your basics are clear, then only you'll be able to make your exam much more fruitful for you. So first let us understand about the protein structure, and there are various categorization. Okay, and these categorization are based on either the biological role, which is also known as function, or the structural role. First we are going to look at the classification of the protein based on biological role. Okay, so first there are structural proteins. So every protein have the structure, so do not confuse it with that. So structural protein has the basic function to provide mechanical support to cells and eventually the organisms. Okay, and it also gives strength to bones, skin, tendons, etc. Right? So this is the basic function that we need to consider, and the example of these are collagen, that you must have heard about, or elastin. Okay, the second type of proteins are enzymes, that we are going to look in much more detail when we come in future lectures. But these are the protein that serves as biological catalysts, that serve as biological catalyst for chemical reactions in cells. So they perform certain function which makes us alive. Okay, the third type of proteins are transport and storage ones. So these are basically our Amazon or Flipkart. Okay, so these are carrier for small molecules. So for example, these are hemoglobin, right? These are lipoproteins or ferritin. Ferritin. So which transport iron in liver. Okay. The fourth type is immune response related protein. Okay, immune proteins I'm going to write. So they are used for defensive purposes. So for example, if your body get infection at that point of time, these protein helps, and you must be knowing about the antibodies, right? So which are IgA, which is also called immunoglobulin A, B, C, not BC, but rather GAMET. Okay, so these are IgA, IgM, IgE, IgD. So this is the short form that you can remember. Okay, and the fifth type is regulatory and receptor proteins. So what happens is there's always cell to cell communication that happens, right? So these type of proteins are called receptor proteins, where a particular cell can communicate with another type of cell, either through chemicals, proteins, or by any other means. Okay, so these are called like regulatory and receptor proteins, and these are basically used in like hormones, DNA binding protein, etc. I hope you get got the idea, right? So let us now understand the protein structure. Now when we are looking for the structure, so like there are multiple level of structure. Okay, so there is a primary, there is secondary, there is a tertiary, and then there's a quaternary structure, right? But before understanding all of these four, first let us understand the protein. Okay, so protein in our body can be globular. What do you mean by globular? So it will exist in what? Spherical or near spherical shape. So these protein are usually soluble in water and they have lots of charged groups. So if you have missed the amino acid lecture, so do watch that. You'll understand why. Okay. So these are very dynamic and flexible. And another type of proteins are fibrous. Okay. So these are elongated and thread-like. So these are not soluble in water. And they have non-polar R group, due to which again these are not soluble in water. And these kind of proteins are usually found in your hair, your nails, as well as your skin. Okay, and these can also be membrane proteins. So we are going to discuss about the lipid biostructure, where we are going to understand, well, like what kind of proteins are there, and what kind of like different lipids actually help in formation or attaching of those particular protein in the membrane. Okay, so I hope you understood this, right? So like you'll find this globular structure of protein or either the fiber structure in your physiology. So now let us understand the primary structure. So what is the primary structure? So this is simply a linear sequence of sequence of amino acids in a protein. So for example, we have studied about the alanine, glycine, leucine, isoleucine, valine, and let's suppose phenylalanine, stuff like that, right? So this structure is called primary structure. Okay, so these are simply linear chain. So if I have to draw a basic structure, so these will be like this. So simply a thread-like structure is your primary structure, right? And the secondary structure. So now what is the secondary structure? So there is the formation of local 3D of the peptide backbone. So there's a formation of some sort of internal structures. Okay, so for example, so this is one amino acid, this is another, this is another, this is another, right? And likewise. So what happens is, so these amino acids are going to form something, right? So that are usually in the form of alpha helix or the beta sheets. I'm going to explain what are the alpha helix beta sheets loops in upcoming lecture, but for now just understand. So protein makes certain type of this structure which is called alpha helix, right? Certain amino acids are capable of making sheet like structure. Okay, so these are called beta sheets. And some amino acid, like I have discussed it with the glycine, so they can form a loop kind of structure. So these are the three basic structure in your secondary structure, right? So when this happens, so this leads to the formation of the tertiary structure. So understand, so in the primary structure there is a simple linear sequence. Okay, So they are in a linear form, right? So there is a formation of some sort of local 3D. So what is happening in local 3D? Either they are forming this helix or the beta sheets or the loops. Okay, due to which there is a global arrangement. Now, what is this global arrangement? So entire sequence of amino acid will have some sort of structure. So either it, even if it is globular, so it will form some sort of that globular structure, or it will form some sort of fibrous structure, or it can have like transmembrane type of structure, right? So there's a global arrangement of secondary structure. So here the side chains are, which is R groups, and other prosthetic groups have a proper 3D structure. Okay, if it is a one single protein, okay, so it will have only the tertiary structure, and if it is a community of the multiple proteins, so it will form the quaternary structures. So what is happening in the quaternary structure is, so like multiple tertiary groups will form. So like there's a tertiary 3D tertiary groups will form to form a quaternary structure. In simple terms, so this is the arrangement of multiple proteins into complexes. Okay, so we have a primary structure which is linear, right? So then we have the secondary structure which comes either in the form of helix or in the form of beta sheets, right? Or in the form of loops or turns, right? Then there's a tertiary structure where we have a complete 3D arrangement of the protein, and then we have a quaternary structure like where multiple of these proteins are associating with each other to form a quaternary structure. I hope you get it right. So in the next class we are going to dive deep, like what kind of structure they make and what are the actual implication of all of this structure. So till then, thank you, bye bye.